that, and neuromuscular conditions. Please give a warm welcome to Dr. Elizabeth Ames.
Awesome. Well, what an amazing introduction, and I hope I live up to it. And also, following Dr. Vockley, I'll do my best to try and explain things that I was asked to. So I'll start off with. I was told if I aim at the back. There we go. Okay, those are my professional disclosures. I'll also say that any viewpoints presented are truly my own and do not represent any sort of institutional position. I will also disclose, because I think it's really important, that I love science. If I have free time, read books about science. I think the most telling thing is that I have named all of my children after scientists. So I'm not an objective person on this regard. Okay, so these were the questions that I was tasked with answering, and I'll do my best to go through them. And I want to start off with talking about why research matters. And I think that that's a question that can mean things to different people, but I think one of the most important things to me is that science is unpredictable. And when we make an advancement, we never know where that's going to come from. And so I wanted to go through a few examples. Does anybody know what this creature is? So it's a Gila monster. Gila monster, so scientists got interested in their spit, their saliva, because it had these unusual properties of regulating blood glucose levels and perhaps being associated with weight loss. And one of the active components in this lizard spit has been the active drug in drugs like Ozempic. So this is an example of how no one would have thought that thinking of a desert lizard spit was going to lead to something so revolutionary. But that's where science takes us, and that's why I think it's so cool. A second example is this really stylized cartoon. So this is a picture of a bacteria, and the bacteria is being attacked by all of these little orange viruses. And it's weird to think about that a bacteria has to have an immune system, but they do. And so about 2 decades ago, a group of scientists decided to set out and study the immune systems of bacteria. And the way bacteria fight off infections is by controlling the DNA. And so that DNA changing machinery is CRISPR. And so this is an example from earlier this year of the first time there was a CRISPR gene therapy designed for one individual. So that individual is the baby KJ. He's with his family and the scientists who developed this medication. And so I think no one would have thought that thinking about the immune system of a bacteria was going to lead to this incredible gene therapy. But that's how science works, and that's why I think it's so cool. All right, and I think we've already alluded to this, right? We have lots of treatments that help us manage disease, but so far we're not really good at curing things like genetic conditions. And so that's what another potential for research is, to truly go after those cures, those longer-term treatments. One example would be spinal muscular atrophy is a genetic condition that sort of affects a baby's muscles and is usually universally lethal by 2 years of age. I think one of the closest things we've come to a cure for a genetic disease is this form of gene therapy called Zolgensma. And so you can see that this little guy is older than 2 years old and is doing great. But we know that not everyone with SMA has this amazing outcome. And so that really still speaks to us that science has work to do. And even in something where it's almost a home run, it's not perfect for everyone. So clinical trials are our avenue for exploring new therapies, like Dr. Vockley was talking about. Like, that's how we understand if these new medications work. And this is a graphic that I took from sort of a think tank website. It's basically saying that rare disease clinical trials are increasing, and it makes me hopeful for the future that we'll have more therapies. And then lastly, science is a team sport, and the best teams include families and patients on their teams. And so, I think being here, sharing your voices, being involved in research is incredibly important. I'll just give a recommendation. One of, I think, the best stories or descriptions of how a drug goes from early experiments to being approved by the FDA was written by Dr. Emil Kakkis, and he talked about how, someone said in the audience, bake sales. So the gentleman on our right, Mark Dant, was doing bake sales to try and create a therapy for his son Ryan, who the book is named after, who had Hurler syndrome or MPS 1. And so it talks about Dr. Kakkis's process of bringing that drug through the FDA approval process. And then the last picture is that Dr. Kakkis is still in touch with Ryan, and that's a picture of when they went to Disney World recently. And so I would recommend this book. It's awesome, and it's a great description of how a drug gets approved. All right, so we've already heard this today, and so I want to give some specific definitions between research and a clinical trial. So research is a general term that describes any sort of study that is meant to get new information. And this could be a lab experiment or an observational natural history study or a clinical trial. A clinical trial is this really specific form of research where a new a new therapy is trialed in a set of patients, and the goal is to understand if it's safe and effective. So all clinical trials are research, but not all forms of research are a clinical trial. And then if you're doing a clinical trial as a researcher, or if you're participating in one, there's a ton of rules, and they're really highly regulated. One of the other questions I was asked to go over is how do you come across a clinical trial? So the best way is a website called clinicaltrials.gov, and I'll talk a little bit more about that. But there's other sources of information that you guys are already using. So, patient advocacy groups, social media. Another great resource is your doctors. So your genetic or your metabolic physicians. Nothing brings me more joy when my patients bring a clinical trial to me or send me one in a portal message and say like, hey, is this something I should do? I love talking about that, and I would hope that everyone's physicians in this room would feel the same way. Okay, so clinicaltrials.gov, for those of you who haven't spent time on it, is a website that is maintained by the US government and is really a registry of all clinical trials being conducted in the US and pretty much all parts of the world. It is like all of NCBI, which is the government's sort of biosciences computing web pages, are going down tonight between 9 p.m. and the weekend. So if you go tomorrow and try and check it, it's not going to be online. So I'm not a liar. They're just doing maintenance. But yeah, so you log on to clinicaltrials.gov, and then you just put in your condition. So one of my favorite FAODs is CPT2, so I put it in. And so you search, and it brings up in the corner a number of studies. So there's 15 studies. It shows the first 10. This is just a screenshot of what it looks like. So some, you can see like have this unknown status. Others have been completed and have results. You can use apply filters to look for the ones that are active and recruiting. So you can use those little click boxes to find the ones that are looking for patients right now. This is a great resource. So if you click into one of these, you can start to get more information. And one of the things I pay attention to is where the clinical trial locations are. If they're all in China or Turkey or some other country, those are probably not the ones to participate in. But certainly if there's one that has a US location, that sort of brings up the prestige and validity of that clinical trial. And then for every entry of a clinical trial, there's a section for contacts. And so that's where you could go to ask about recruitment and participating. There's usually a contact like number or email for patients, and then also for healthcare providers. So if you don't feel comfortable contacting this trial about participating, you could ask your physician to reach out on your behalf. I do that all the time. So this is another really great resource for getting involved in clinical trials. Okay. And we've heard these phrases already today, and so I'm going to sort of do a 30,000-foot view of the clinical trial process. So it starts with preclinical data before any human is ever involved. Then there's phase 1, 2, and 3. That's really when humans are being used for the subjects. And then lastly, sometimes there's phase 4 that I can talk about at the end. Okay, so this is, and we've already heard about like the different mouse models, so there's so much like foreshadowing or like crosstalk that this is great. So these are all of the examples of mouse models for FAODs that I took from one of Dr. Gillingham's publications. So it's nicely laying out that there's a ton of mouse models, and we heard from Dr. Vockley's talk that they're incredibly useful in starting to understand if a drug or a therapy might work and if it's worth moving into humans. I think mice get a lot of attention as being like this model for doing science, but there's other animal studies that are equally important. I love dogs, so I'm gonna do dog examples. So this x-ray is showing at the end of, I guess, the dog's foreleg that there's this really aggressive bone tumor called osteosarcoma. Osteosarcomas are common in certain types of dog breeds, so particularly Rottweilers. And so because this is a naturally occurring model of this really aggressive cancer, it's incredibly helpful in studying whether a cancer therapy works. And so because these dogs get osteosarcomas and we're such a great animal model, it has led to revolutionary discoveries in treating cancers. So IL-2 immunotherapy, CAR T or cell therapy, checkpoint inhibitors, and cancer vaccines all at some point were done in dogs before they transitioned into human studies. All right, I told you I love dogs. So this is my second example of how animal models can be really helpful. So this golden retriever is special because he has Duchenne muscular dystrophy. You can sort of appreciate that his back end is maybe a little bit smaller and atrophied compared to a regular golden. And so Goldens served as this really helpful animal model for us understanding how to treat Duchenne muscular dystrophy. And so that has meant that we understood that there was maybe benefit for things like exon skipping therapies and gene therapies, but we also learned that it didn't seem like stem cell therapies were as helpful in this dog model. And so it was really important to know before these types of therapies were tried in humans. Okay, so those were all examples of preclinical, so animal models before humans were involved. Generally speaking, phase 1 is just looking at safety. And so, you know, if it's a drug for high cholesterol, you would do it in a healthy subject, but for rare diseases, oftentimes phase 1 and 2 are combined. And so phase 2 is sort of looking for early signs that the drug is working. And then phase 3 is a larger study. So like we heard with Dr. Vockley, he's looking for more subjects in phase 3 trials to really understand if there's a benefit for these new therapies. After a drug goes through a phase 3 trial, the investigators or the biotech or pharma company packages up all of this information and then brings it to the FDA. And then it's the FDA's job to review all that data to decide whether or not the drug is approved for use widely in humans. And then phase 4 is a post-approval monitoring. So for certainly cell therapies like these CAR T therapies, they require a 15-year follow-up period, which is incredibly long, is expensive, but has been really important for us understanding that there are cancer risks for these types of therapies after many years. And so phase 4 trials, especially for new forms of therapies like gene therapy, cell therapy, is common. Okay, one of the other questions I was tasked with answering is, why do trials get canceled? So sometimes it's not enough patients, which is common in rare diseases. Sometimes funding runs out. Sometimes there are safety concerns or unexpected side effects. So there have been a number of gene therapy trials that have been shut down because there was a death. And any death is tragic, but then it's also sort of doubly tragic for the rest of the community who was hoping that might be a treatment. But it is a common reason for trials to get canceled when it's something like a gene therapy trial. And then oftentimes, the overwhelming majority of these drug trials are conducted by biotech or pharma companies. And so they're these huge corporations, or else they're a really small startup, and they go through all these rapid changes. So companies can restructure, they can change their priorities and just shut down a trial, even if it looked like it was going to be promising. In terms of doing clinical trials for rare diseases, there's a couple other considerations. I've already mentioned it's really hard because there's just fewer patients. One other consideration is that in order to prove if a drug works, you have to have a really good understanding of what the disease looks like untreated. That's why natural history trials are so important, because it lets us understand sort of how a person with a condition goes through life and what health concerns they experience along the way. And so some of the trials that I've participated in for other rare diseases realize as they're starting the trial that they don't have enough natural history data. And so then simultaneously they're trying to collect natural history data in addition to their trial data. And it sometimes doesn't work out just because you do need that time and that experience to understand the disease. For the gene therapy for SMA, that was incredibly important because we had a natural history that clearly showed the health concerns and the lifespan of babies and infants with SMA. And that's what that was a critical part of the FDA approving that therapy. And then one other thing that I was sort of initially titled this slide challenges, but I think is more of a consideration, is oftentimes rare disease trials are adaptive. So if it's a gene therapy trial and they're doing dosing, they oftentimes will dose a cohort, wait, watch how they do, review the safety data, and then decide if they're going to increase the dose or maybe stay the course. So that's a unique feature of rare disease trials that I think is actually great, is that they're responding to how the patients are doing. All right, so this is a very simple slide for something that is incredibly complicated. But one of the questions that I was asked to discuss is how research gets funded. There's lots of different ways that you can look for money. The National Institutes of Health, or the NIH, is one of the most important funders of research in our country. And it's really grants that are given to academic researchers. Additionally, there are private foundations, patient advocacy or crowdfunding, and then obviously biotech companies will like getting involved when a drug is near to FDA approval, because that's when they think that they can, swoop in and make the most money. But the NIH is really important for those early stage research projects. So how does a grant work? So at the beginning, it involves researchers sitting down and coming up with this really, really intricate, really detailed proposal of a series of experiments that they think will answer an important question. And there's different combinations of letters and numbers, and that describes how long or how much money the grant is worth, so R01 versus an R21. Once that grant is submitted, it's peer reviewed by a group of scientists called a study section. That study section is oftentimes senior scientists who, they themselves are successful at getting grants. And they look through all of the grants that were submitted on a certain topic or in a certain field, and they judge the proposal based on science, innovation, and the team. Can the people proposing the research actually get it done? So they go through and they talk about them and they score them, and then the NIH has funding lines, and so if a grant is scored above the funding line, then in general it's a funded grant. If a grant is funded, the money is often split into two different categories. The direct costs, so exactly how much it costs to buy the mice, to pay the technician, like the consumables that you can clearly write a line on the budget. But then there's also money from a grant that goes to indirect costs. And indirect costs are the infrastructure, electricity, the research administrative staff that moves the grant or moves the actual money from the NIH to the institution, the people who do the IRB to make sure that the research is being done ethically. Those are all expenses that are really hard to capture on a line in the budget. And I think earlier this year there was a lot of discussion about this current administration capping indirect costs at a relatively low percentage. And the way that it was discussed is that they said that indirect costs were wasted money, but it really is important to pay for things like electricity and DNA sequencing cores, and it's just really hard to know exactly how much to put on a budget line, and so that's why it's sort of considered this indirect or cost. And simply put, if without indirect costs, it is very, very challenging for an institution, a university, to actually get that research done. In general, the NIH caps are based on an institution negotiations with the NIH, and so they can actually vary widely between 8% and then 55%. And then to sort of finish off the discussion about the NIH and grant funding, the question is, well, gosh, in the grand scheme of things, why is the NIH funding important? It is truly the most important key source of early stage research. It is one of the only places where you could propose to study the immune system of a bacteria and potentially get that funded. When the budget is cut, it really does freeze projects, and it is incredibly challenging to finish a series of experiments if the money is just automatically taken away. I read that a really good, I think, description of, if a grant is stopped, if it's a $5 million grant and the researchers have done $4 million worth of work, that, freezing or, canceling that grant isn't saving the American public $1 million. It's really wasting $4 million because maybe they just needed 2 more sets of mice to prove that their experiments worked. And so that's been a really big challenge this year for researchers. And then I think simply, if there's less NIH funding, it means less breakthroughs, less chances to come up with some new revolutionary finding, because science is unpredictable. I also want to point out that beyond, the scientific discoveries, the NIH funding is an incredibly good investment. I'm not like a huge money person, but if you calculate the return on investment, every dollar that's spent on the NIH generates $2.56 for our economy. It's giving people jobs. It's driving these startup companies that might come up with this really big blockbuster drug. So it does have benefit just beyond doing science. Okay, I'm going to shift gears and tackle another set of questions that was posed to me. And so that has to do with sort of the structure of the federal government. So when you think about the Department of Health and Human Services, there's 13 operating divisions. We've talked about 2 of them, the NIH and the FDA. But sometimes there's confusion about who does what. So the NIH, as we talked about, funds and conducts research. It decides what science to support, but it doesn't do anything with drug approvals. In contrast, the FDA is the government agency that's meant to approve drugs for use in the United States. They do that by reviewing safety and efficacy or effectiveness, and sometimes they either approve it, can request for more trials, or deny the approval. So the goals of the FDA are to protect patients and to advance therapies. That means ensuring safety and effectiveness for medications, vaccines, medical devices, and now things like cell and gene therapy. One important thing in the sort of history of the FDA is that the Orphan Drug Act that was passed in 1983 allowed for more approvals for rare diseases because they understood that we were never going to be able to do a trial with thousands of patients. And then during that FDA drug approval process, they do ask for family input, and so that is another important piece as well. I put this picture up to talk about the sort of history and the importance of the FDA. So in the 1950s and 60s, there was this morning sickness medicine that was used in Europe called thalidomide. And the drug makers of thalidomide approached the FDA and said, oh, we would like to sell this medicine in the US. And it was because of one reviewer at the FDA being really careful and really thorough, noticed that children who were born to moms who took thalidomide had these pretty striking birth defects involving their limbs. And so because that one person at the FDA was so careful, I mean, she really did save a generation of Americans from these really profound birth defects. So the FDA definitely is something that is meant to protect us. Okay, I'm gonna switch gears again and talk about one other agency. So the Health Resources and Services Administration, or HRSA. So HRSA is not a household name, but one of the things it's involved in is the federal, so US government level, oversight of newborn screening. So taking a step back, so the part of the role of HRSA is to come up with a recommended uniform screening panel, or the RUSP. And that is a series of core conditions that they recommend every state should screen babies for. Another sort of vegetable or like alphabet soup term to know is the ACT DNC, or the Advisory Committee on Heritable Disorders in Newborns or Children. That is a body of volunteer experts who review the data for a new disorder and decide whether or not it should be included on the RUS.